esterase inhibitors. He has published 128 international peer-reviewed papers with H-index of 24 and number of citations 1,500 in Google Scholar. H-index of 20 and number of citations in 1,160 in Scopus, with a cumulative impact factor more than 260 according to JCR reports of 2020. He has authored a book, 5 books, chapters, and edited a book in Springer. He presented 23 invited talks at national level. In 2019, he was an invited speaker in International Multidisciplinary Symposium on Drug Research and Development at İnönü University, Malatya, Turkey. He is carrying out to the design of spike protein binders for COVID-19, serum albumin interaction studies, and designing new molecules for human colon cancer using HTC 116 cell lines. He received 3 research funds at national and also at international funding too. In 2019, his 2 research papers were awarded as the most accessed papers in Wiley publications. He is currently guiding 5 PhD students and appointed as PhD thesis evaluator in University of KwaZulu-Natal, South Africa. He is a chief executive guest editor of the journals of Current Drug Targets and Combinatorial Chemistry and High Throughput Screening. He is a section editor of Journal of Coronaviruses in Bentham Science. He received a patent from Korean Intellectual Property Office in the topic of protective and or therapeutic compositions of neurological disorders containing furanochalcones as effective agents. Recently, he filed 1 more patent in the development of composition for preventing and treating neurological disorders. Ethyl acetohydroxamate incorporated chalcones as an active ingredient. With this introduction, I would like to hand over the session to Bijo sir. Bijo sir, the session is yours.
So thank you very much. So is it audible? Is it audible to all?
Bijo, you are audible.
Yeah. Can you see the front slide of my PPT?
Yes, sir, it's visible.
Is it visible to all? Hello?
Yes, sir, it's visible, sir.
Suppose if you are facing any problem, you can ask.
Okay, sir.
Hello?
Yes, okay, sir.
Okay. So thank you, Nirmala College of Pharmacy, especially the Department of Pharmacology, for inviting this particular area, especially the drug repositioning. So actually recently only I started my research in this particular area. So actually the department gave me 1 hour to finish the program. So previously I know that it is very difficult to finish all the aspects of drug repositioning within this small period. Anyway, so what is the core concept? Please, hello, please mute your audios. Hello.
Participants, please mute your audios. Mute
So
your
some
mics.
of the audio. Okay, otherwise it will interrupt my lecture. Okay. I think this particular drug repositioning, I think past 20 years, this particular concept was established frequently in the field of medicinal chemistry. But I think during the entry of COVID-19 only, we saw the well of the very famous drugs, something like HCQ, chloroquine, which are belongs to this particular category, especially the drug reposition. But still there is a conflict of reports are still there. The use of that particular drugs. Okay, I'm not going to in that particular area. So we will see the what is a real concept of drug repositioning. Okay, I see here, I think I used a particular term. Basically, it is a hard work into a smart work. Okay, I think if you are evaluating my slides, you can clearly understand what I mentioned in my first slide. Okay, clear? Now we will slowly go to the, can you see the next slide?
Yes, sir. It's coming.
Okay. So now I think this is a term we can tell either in the drug repositioning or repurposing. Also, in some of the scientists, they are also using repurposing, repositioning. So both are same only. I think in some of the research core concept, they will also tell it is drug rescue. So that is another word also, drug repositioning. And also you can tell that it is a drug reprofiling and also drug retasking. Okay, so many synonyms are available for this particular area: drug rescue, reprofiling, and drug retasking. Okay, so the concepts of all these words are the same only. So you can see that what is a major core concept. So existing of a drug for a new indication. Okay, so already you have a established drug for a particular disease and its own pharmacology and its own ADME profile, all the things we know that. But the concept is here, we are going to implement your drug molecule or that particular formulation or that particular dosage form and into another therapeutic indication. That's why the definition is, it is an existing of drug which is entirely for a new indication. Okay, so that is a first slide. And then we will see that what I think I will show one of the very famous picture. So then we can clearly see that what is the concept I'm going to share. Okay, so this is a transformation of hard work into smart work. Okay, why I used this particular term? So you know that hard work. So in hard work, if you are concerning about the drug discovery process from a synthesis to the development of a molecule or the synthesis of a molecule from starting to the FDA approved drug, there is a huge, enormous amount of work has been takes place. Something synthesis, biological screening, phase 1, phase 2, phase 3, phase 4. So many hurdles you can see that. Okay, so there are many things we can see for this the transformation of hard work. Suppose in case of your drug repositioning, so we already you have an established hard work is there. So that hard work we are slightly changing into a smart work. Okay, I will demonstrate how it is happened. So this is a simple demonstration. You can see that you see here you can see this graphical presentation itself. It is clear that what I mentioned. Okay, so hard work. Okay, so by you see here in the case of your repositioning, we are changing the object in a different manner. So and finally you will reach your goal as soon as early. So that is a hard work and smart work, the difference between hard work and smart work. Okay, now we will go to the next slide. This slide, I'm just comparing the drug development in a traditional way and how the traditional way is entirely different from the drug reposition. Okay, I think already you have a slight idea about the traditional concept of drug design. The traditional concept of drug design majorly connecting with the identification of a target, identification of the disease, and the synthesis of your molecule. That is the most important one, the designing and synthesis of the molecule. Once you Hello. Can you hear the voice?
Yes, sir, it's audible.
Okay. So now in the case of a traditional drug tracing concept, I think this pictorial diagram itself, it gives a clear factor. You see here in a traditional concept, we are passing a huge amount of research has been takes place from identifying the therapeutic target, and finally it will launch to the clinical approved drugs. You see here, once it is approved for a clinically established drug for a molecule, so we are typically concentrating on that particular molecule in that corresponding disease only. But in the case of your repositioning, so we are making a bypass. So this drug can be entirely, it can be used for another areas, and we will see that you see here what is the advantage of this particular concept is by making this particular shortcut. So you can see that there should be a pre-approved drugs are you are handling, so there is no problem with the toxicity, because already we know the toxicity profile of your molecule. So already we know that which is the best dosage form it is available, and already we know that the IC50 value and inhibition constant, effective concentration, all the biochemical parameters you can you know concerning about the drug molecule in for your old disease. But once you are transforming into your new area, so by analyzing these preliminary information, so we are slightly going to the I think there's no need for go for. So we are slightly going for the drug repositioning in the area of phase 1, phase 2, phase 3, and finally you can go for your final clinical approval. So this is a simple diagrammatic approach. I think we will slowly see the concept of, because the concept is very simple, that we are taking a particular drug molecule, the old drug molecule, we are repurposing a new therapeutic indication. So slowly we will see that how these things can be happening, and what are the hurdles you should cross during the time of your repositioning. Okay, I think the remaining slides you can see a clear idea about that. So this is a you see here by seeing this particular concept, I think everyone have a clear mind that so why it is very simple. You see here, so already we have a drug molecule, we have a target, but you are transforming the target from one to another. So by in a theoretical way, it is a very simple conceptually, but it is not simpler. That's why I just make a statement. Drug reposition, it's a very simple conceptually, but not simpler. Okay, we will see that how it will happen. Okay, I will demonstrate one of the very common concept of pharmacology. I'm taking a drug molecule A. Okay, so I have a prescribed, okay, a established structure. So I have some good indication of my structure, all the things I have. I developed a particular molecule of A. You please see this. So this particular A molecule, I am just concentrating target 1, target 2, target 3. You see here, there are many targets are there. So don't think that suppose if you are administering a particular molecule, it will target only one. Definitely it will target some. It will also, because there's a target on is there, target off is there. Okay, there are many mechanisms are there. Anyway, you just in this graph, I just representation I make. So suppose imagine there are 3 targets which is responsible to interact with your A drug molecule. In that the eventually you will get a disease X. For the disease treatment, we are employing a drug molecule A. Okay, so now I'm just taking another drug molecule, the same conceptually. I'm just taking a drug molecule B. Here also the same type of mechanism I can explain. Here also the target may not be same, it may be different, it may be in a different system also. So it may be in a different type also. Okay, so this is target A, target B, target C. So here also you can expect so many targets. So I think it may be knocked down any one of them, or it may be acting on multiple. So that is depends upon your drug molecule. So here also you will it is available for a disease Y. You see here, in these two concepts, I have a clear idea that I have a drug molecule, and there are 3 phases. I can see that a drug molecule, targets, and disease. in case of your second also a drug molecule, targets and disease. So please, the clear fact is that there is no correlation between the structure of drug A and drug B. There is no correlation between the target 1 and target A. Maybe there is no correlation between the disease X and Y. Can you hear the voice clearly? Okay.
Yes, sir. You're audible, sir.
So sometimes you see here, I cannot see your faces because normally we are practicing only normal lecture classes. So I'm not comfort with the webinar. So that is practically I'm speaking. Okay. Okay. Now you see here, we saw a general concept. Now by making a simple shortcut, I will show you how the reposition can become. So you see here, this is a concept. I am making a repositioning. Drug A has a tendency to reach the target A. So preliminary, we identified or we established the drug molecule on the basis of treating the disease X. Now the total scenario is changes. So now you can see that the drug A, I just concentrated for the target A, which is meant for the disease Y. This is, you can tell that the reposition basic principle. Okay, so once if you know this concept, obviously we will see that how this repositioning can become with the basic principles. I will tell the basic principle. Okay, we will go to the next slide. So we know that, I think obviously you should have a mind that there are there are n number of not n number of lakhs of molecules are synthesized in our labs. There are many molecules are in the chemical abstract. So unfortunately only I think 1600 FDA approved molecules are available, and also some of the molecules are not available in the rare diseases or also neglected disease. Also you cannot see much amount of drug molecule. So that means the library of FDA approved drugs are very small only. So within that small library only, you should develop for a new disease. Okay, now before going that particular, that's why the term is why the drug before the drug reposition. Suppose if you are a researcher, so before the drug reposition, what are the things you should be noticed preliminarily? That is very important. Okay, please see that. Identification of a rare disease with the lack of promising drugs. We know that SARS-CoV and COVID-19 related, you know that it is very difficult. I think 2016 to 17 only the that particular enzyme was also established. I think in the year of 2020 only the exact genomes of the that particular target was established. So that's why this is not a common disease, because suddenly it happened. So that's why identification of a rare disease with the lack of, suppose if you are facing a diabetic or cancer or hypertension, so very commonly disease, we have an established molecule. There is no issues connecting with the either in the chemist or either the medical chemist or the clinicians. There is no problem. But if you are facing a rare disease with a lack of promising drugs, so there is a chance for a new drug molecule or a old drug molecule with a new target. So that's why the scenario will comes in like that way. So once you are identifying a rare disease, next the concept is this is the most important one. You see here, that's why I initially I told you know that. So if I will explain all the things, I will never get time to complete within this prescribed time. So anyway, I will tell that. So once you identify the rare disease, the identification of the pathway which will lead to the disease is very, very important, because in every disease there is a hidden pathway or a established pathway is necessary which will lead to the disease. So we should know the pathway which will lead to the disease. That is very important. So once you are identifying, the next phase, repurposing a drug by in silico, okay, and knowledge based. Okay, actually, so there are two types of drug repurposing, mainly in silico. So actually, my research area is a in silico based method. So sometimes I will also use the knowledge based also. It's a mixed methods, because majorly I'm using the in silico method for the repurposing the molecule. So that is the we will slowly see that what is in silico type of method and what is knowledge based methods. Okay. We will see that. Now you see here, what is the advantage of the old drug means you have a safety profile and you know the side effects. Okay, suppose if you design a particular drug molecule, a target, you have a clear idea about the safety of your drug molecule and the side effect of your drug molecules. So all the preliminary investigations, you can see that of your particular drug molecule. So then only you are going for your repurposing. Okay, now we will see the conclusion of this concept. The conclusion is a reduced requirement of the early clinical stage. Okay, you know that because before the clinical establishment, there is a huge amount of synthesis, formulation studies, enzyme inhibition studies, and many things are available. And preclinical testing, in vitro evaluation. So and such type of things you can reduce it in the early stage of clinical stage. That's why I used the term is drug repositioning. Okay, this is the primary concept we should go for before the drug repositioning. Now we will see the next slide. What is the road of drug repositioning? Okay, the road is very, very important. And what are the phases is drug repositioning? Okay, this is the first thing. Before establishing a drug molecule for a new target, you should have a clear idea about the adverse effects. What type of adverse effects commonly it can produce in a particular biological scenario. That is the one of the major thing you should notice, the adverse effect of your current drug molecule. Now, the gene expression. The gene expression, that is a different story. So the gene expression, once you are identifying the adverse effects of a particular drug molecule, so you should know that in what type of gene expression which is responsible to trigger your molecule for a particular target. Okay. So maybe the gene expression can become in many ways. So you should know the gene expression of your drug target. It's very, very important. Okay. And the pathway identification, the same thing already I mentioned it in my past slide itself. So these are the first preliminary investigation you should know about. So I can tell that by seeing this first road itself, you should have a clear idea about your drug molecule and the disease. Your preliminary investigation should be your adverse effect, gene expression, and pathway of the disease. All the things you should know that in the first road. At the second phase, we will see that the similarity searched ligands. That is the thing I am because normally my research group, we are employing this type of method. So we don't have any type of knowledge-based method for existing knowledge. Suppose because as a medicinal chemist, what is the thing we are going to explore means, suppose if we have a structure means on the basis of the similarity-based screening, we can easily predict that. So suppose what type of pharmacological activity it can produce for this particular target. So this is very important. The similarity search ligands. There are many types of both commercial and academic softwares are there. So today I will show a commercial based software and how the 3D and 2D concept of ligands can be identified by the means of similarity search method. Similarity search method is a concept is the similar drug molecules which can recognize a similar type of receptor. Okay, so that is I think we know everyone know these particular concepts. Okay, there is no confusion in that. Now the literature mining, you see here, so without literature mining, you cannot establish. Okay, so the literature mining, it should involve the chemistry of your molecule and the generation, the chemistry of your, not the chemistry of your existing molecule and the pathophysiology of your new molecule and what type of relation you can correlate between your existing drug and the new disease. That's why the literature mining, which is connecting with the new drug development, is one of the very important area. Literature mining is very important. Okay, now you will get a drug reposition from a bright idea. Okay, you see here, I think by seeing these slides, I think everyone will say that, okay, maybe it's a very simple task only. Okay, I will show that in the remaining slide, you can see that what type of hurdles we are facing the drug repositioning. Okay, normally we can make a drug molecule, and you see here, if you're making a drug molecule, I told you know that from IND application to the FDA application, it will hurdle, it will process many more process. Okay, so such type of process can be bypassed by this method. But anyway, these also have a n number of hurdles. Okay, we will see that individually. What are the things we are facing? Okay. Now we will slowly. What is a necessity of drug repositioning? That is, you see here already. I told that it should be. You see, the previous slide itself. I told rare and neglected disease. Okay, suppose if you are designing or if you are seeing a drug molecule, so something like diabetic or hypertension or cancer, you see here that is not a rare disease, it is a common disease. But if you are experiencing a rare or neglected disease, why the pharmaceutical companies have much more awareness of, they will never have much more awareness of this particular area, because what is the thing is, I will explain, it's a very small population, and profit may be very less. Okay, so so you see here the pharmaceutical companies, they are aiming for only profit. Okay, so they are maintaining the chemist for the designing of new molecules, and their main aim is to make promising molecules with huge amount of profit. So the profit is one of the major aspects of pharmaceutical company. That is a different story. I'm not going to explore the thing, but anyway, the small population pharmaceutical company will always will never have such type of huge interest. Something like the other disease. So the rare and neglected disease always have a small population. Though now the finding at that particular time, so they will never have a time to investigate more time to for the development of a new drug molecule. They will try to make a new therapy with the existing drug itself. Okay, so that's why I told that in the initial time of the pandemic situation of COVID-19, you see here the Lancet journal itself published hydroxychloroquine was one of the major drug molecules can be used as a drug repositioning. So after so many of the studies from the France and something like from the USA, finally they published that it is not an effective one. Maybe it's a prophylaxis. Okay, so that's why you see here such type of things. I will show in the remaining slide how that's why finding a new drugs with the existing drug is the most easiest one for a pharmaceutical company. So they need to clarify some of the approval agencies also. That is the only thing, and they should get the clinical trials also. So that's why approving existing drug, it is very easiest one for at this particular situation. Okay, now the question is finding a new use for a withdrawn drug also. Okay, so actually my core area is also, I am also working on the drug molecule which is withdrawing from a particular area. So that is also I am trying also. Okay, sometimes if you will get good results, sometimes you will not be get. So that is depends upon the nature of your research, what you are going to employ. So you can also employ the withdrawn drug also, and also you can modify the drug also. So anything you can see that is typically on the convenience of a medicinal chemist. Okay, so these are the necessity of the reposition. The conclusion is very simple. As a very famous example, I think the pharmacy students, we know the very famous example is thalidomide, which is a very famous drug for the treatment of morning sickness. But unfortunately, so due to the constant fetus problem, that particular drug was it was withdrawn by the US FDA. So because due to the dangerous side effect. But the thing is, the dangerous drug, the fact is a very curious one. So thalidomide, so later it is employed for the treatment of multiple myeloma. That is real magic of drug reposition. So a drug was withdrawn due to a severe problems of a pregnant woman. So unfortunately, so the US FDA drug is banned that particular drug molecule, but there may be a chance. You see, the drug reposition can becomes in a way of multiple myeloma. So that is established. I think, and another very, I think the most, I think the next one is, I think very, what is that? Already that is Viagra. You see, that is Lindafil. It is a major drug for the angina. Okay, but you see here, it's adverse effect, which will lead to the erectile function. Now it is a Viagra. You know that the most profitable area, I think it's Pfizer only. I think this particular drug was, I think the major patent went for that Pfizer only. Okay, so this is you see a drug reposition of the very famous example, that is the erectile dysfunction, a drug which is originated because the original drug is responsible to treat the angina pectoris. So unfortunately, so there may be some problems arise from that. So it will launch to a new area, so which is used in the major problem of erectile dysfunction. Okay, now we will see another. A bit maybe less. Okay, fine. Can you see the thalidomide withdrawn? Can you see that?
Continue, sir. Continue, sir.
Okay. So now I told you know that the final one is the another. This is a now we are slowly comes to the importance of medicinal chemistry. You see here, sorry, the time is. Suppose if you're getting bored, you please inform me. Okay, because actually your that department gave me only 1 hour. Okay. So anyway, I will try to finish as soon as early. Okay, now we will see that drug repositioning, a new perspective of a medical chemist mind. This mind is very, very important for a medical chemist for designing, or because his perspective mind is one of the very important thing. We will see that. Can you see the slides now with its demonstration also? Can you see?
Yes, sir. Yeah, it's all visible, sir.
Yeah. Okay. So you see, this is my area. I already told you know that I'm working on the FDA approved drugs. At the same time, I am working on the withdrawn drug also. So withdrawn drug, don't think that withdrawn drugs doesn't have. There may be some magical property may be present in that. So as a medical chemistry point of view, you should see that how this particular drug can be effectively used for another target. Okay, so by this you can see that what are the, suppose if you are a researcher for a drug repositioning, so this is my concept of drug repositioning. What are the things you should be known? So suppose if you analyze the FDA approved drug or withdrawn drugs, so you please note that the collection of raw data, both drug and the targets, that is necessary. That's why I already told you know that literature mining is very, very important. Now can you see now the animations clearly?
Yes, sir. Yes, it's clear.
Yeah.
Yes, sir,
Now
clear.
we will see. You see here. This is the most. Please see these slides, because these slides you see here, you cannot see these slides from any of the SlideShare or in the Google, because all the concepts are new only. So this is a very important concept. You can see that once you are collecting the drug targets, you please see the first thing is you have new drug target with all the therapeutic indication. That is the first view. You can see that a new target with the old therapeutic indication. You can also an old target with the new therapeutic indication. You see here a new target with old therapeutic indication, an old target with the new therapeutic indication, and another. Again, you will get another idea, because all these ideas I got from the collection of the raw data for the target. A new target with a new therapeutic indication. See, can you see the difference clearly? Hello, anyone, please respond. That's why you know that webinar actually I cannot see the face of my candidates. So if I
Yes, sir. It's visible, sir.
okay, yeah. Did you understand clearly?
Yes, sir.
Yes, sir.
Yeah, you see here, once you are identifying raw data from your withdrawn or FDA approved drugs, a chemist mind should have three things. One is new target with the old one, new target with the new therapeutic indication, and old target with the new therapeutic indication. Now you please accumulate all your data mining. You will see an evidence-based in silico. So that is my area. Okay, only. So we will see that then proof in experimental, that is also very, very important. Okay, and the clinical development of FDA approved drug. Okay, so these are the perspective area. The perspective area is the middle portion, the portion which is I mentioned in the yellow. So can you see the portions I mentioned in the yellow? Okay, you see here. 1 minute. Pointer option is there. Okay. So this area, this is a very important thing. New target with old therapeutic indication, new target with new therapeutic indication, old target with new therapeutic. So you please see this. This is very, very important. Okay, so before beginning the drug reposition of your research, so you should note that which is your core area. Is it for the new old target with the new, or new target with the new, or the new target with the old? That is important. Okay, please take care in your mind. Now, finally, development of your drug repurposing. Okay, the short form is DR. Okay. Now, the conclusion of this slide: drug discovery and the clinical development. And now, the post-marketing surveillance. All these things you can see from our previous publication. I will show at the end of the slide. You can see a detailed way. You can see that. And the drug repositioning. See, this is a simple concept of drug repositioning. The 4 phases. Okay. Now we will see that the various approaches of repositioning. Can you see the slide clearly? Hello?
Yes, sir, it's visible, sir.
Okay. So, because actually I don't know that sometimes. Okay. Now we will see that the approaches of drug repositioning. Okay. Is now Prashant sir, this time is over or not?
No, sir. Please continue, sir. No problem.
Okay, please give me some more time. Okay.
Sure, sir.
So various approaches, various approaches of drug repositioning. Okay, so I will finish. So first one is the in silico method. That is the most accepted method nowadays. I will show one of the methods which is used in my lab. And the second one is the biological method and models which is developed on the base of the molecular pathway. And the third method is the experimental method. I think you see the definition, the target screening cell assay, animal model, clinical trial. That is another way of drug repositioning methods. And this is a mixed method. Actually, I used the first method and the fourth method. I used both in silico and mixed method, computation and biological experiments. So, which will strengthen your drug repositioning, concept in a better way. That is clinical testing before the clinical testing. Okay. So now the last is the knowledge based. So basically knowledge based drug repositioning, commonly it will not be handled by the medicinal chemist, normally handled by the expert physician. Okay. Okay, they only have the knowledge base. Okay, some researchers also employing, but anyway, the doctors are capability of interpreting the results. I will show that how the knowledge based the drug repositioning will comes in a very successful way. Okay, these are the 5 concepts: in silico, biological, experimental, mixed knowledge. Okay, so you see here the last concept. You see here, I think 95% of the participants are, I think, the pharmacy phased. So I think this particular area is very difficult. I think maybe the clinical people or those who finish it, or for those who have PharmD area, so they can go for the knowledge-based drug repositioning. So there is no issue in that. But majorly, the ADR monitoring, all the things comes in the area of doctors. That's why I mentioned it is 5 only. So actually accidentally mistake, the typing mistake only. This is not 4. This is 5 only. Okay, sorry for that. Now we will see the next slide. Okay, the in silico, because the remaining methods, you know that I cannot finish within this prescribed time. So I will cut short my area into the in silico, text mining, cluster propagation, semantic and database resources. We will see the last one, the database and the cluster based, we will see in the last one. Okay, now in silico approaches. The first in silico text mining. Very important. Mine the information and collection. Okay, so there are various sources are there. Resources are available for identification of a drug molecule, PubMed database, drug bank, ChemPaper, and ChemSpider, SciFinder. So many data mining resources are there for getting the information of your molecule. Okay, now the next is cluster. Cluster is nothing but very important. There should be an established relation between the drug and target. And next is propagation and disease to gene relation and disease to disease relation, because this is a first disease and the second disease, the new therapeutic indication. You please make a clear idea about the disease relation. and target disease relation, disease gene relation, disease relation, and target disease relation. Okay, these are the major things can becomes in the in silico approaches once we are going for a propagation method. Now the next is the semantics and the new algorithm. So this is a very you see here I think the peoples who are working on bioinformatics, or the persons who finished their MPharm in pharmaceutical chemistry, this is the most what is that promising area. I think unfortunately our pharmacy syllabus doesn't have to focus on this particular area. We are just studying the conventional concept of the synthesis of aspirin and indomethacin. There is no use in that modern era of chemistry. So this is a new concept. The semantic approaches, the new algorithms developed basis on the ontology of the network. So there should be a bioinformatics also needed to satisfy the what is that in our pharmacy syllabus. Very important. I think future it will become. Okay. So these type of newer techniques, it will give more chance to the students to enroll in a different way of research. Okay, I think nowadays all these particular development of algorithms and other software designs are handled by the bioinformatics people. But anyways, in some of the peoples, if a person know both chemistry and algorithms, so definitely he can make a successful. But in what is our problem is we know the best design. Unfortunately, we don't know the algorithm. So suppose if person know the algorithm, he don't know to design a molecule. So if a person know algorithm and design, he may not be knowing any knowledge about the synthesis. You see here, all these things, three ways should become. Then only you can design and execute a molecule. Okay, no need to tell more. Okay, this is a database that is pharmacological and chemical and proteomics area. That is also another what is that documentation part for the in silico approaches. Okay, clear. Can you see the slides clearly?
Yes, sir. It's
Yeah,
visible, sir.
I will share my slides. Sorry, I will share my slides at the end of the. I think you will receive this soft copy. You will receive. No issue in that. Okay, now I think we are moving to. Don't worry about that. I think the participants are getting bored. I think more than I took 1 hour. Please don't mind that. Okay, now we will see 1 or 2 examples. Then we can finalize the your webinar. Okay, now the drug combination with the new drugs with the old one. We will take some examples. Okay, can you see the name of? Can you see the structure? It can be target on the EGFR. I think it is a very famous drug of erlotinib. And this is a kinase inhibitor. Maybe it is not included in the syllabus, but anyway, I think MPharm people can know this particular drug molecule. This is used for. Can you see the abbreviation? The abbreviation is very common. Can you do you know that abbreviation? It is used for the treatment of non-small cell lung cancer. Okay, recently I wrote a book for the a book, not a book chapter for this particular area, especially the EGFR. Okay, I will share that. You can get more ideas about this. Basically, it is a kinosol nucleus. And what is the problem of this particular drug? Prolonged use, it will resist your cancer cells. Okay, so basically it is a quinozoline with a pyridine with a acetylene link, acetylene side chain. But what is you see here? Long use, it may be resist your cancer cells. So what is the thing? The heading is what? Combination of a new drug with the old drug with a new therapeutic indication. Sorry, old therapeutic indication. You will get the drug reposition. I think this structure is, I think everyone, chloroquine, and why if you are administrating this particular kinase inhibitor, it will work for, and hydroxychloroquine also can be employed. This is a very classical example of the combination of new drug with the old one. Okay, now we go to the next example, knowledge based. This is, I think this is very common for all the very famous structure of metformin. Basically, it's a biguanide derivative. So we will see that what type of drug repositions are currently available. Okay, you see here, basically it is a biguanide metformin. Type 2 diabetes, very famous drug, type 2 diabetes. What is the problem? You see here, 1,500, 1,000 gram, not 1,000 milligram or 500 milligram. You see here, huge amount of drug molecule, amount of drug should be needed for the therapeutic action. So now the concept is metformin consumed patients with a lower risk of breast cancer. This is a typical based knowledge, or it is a knowledge based drug repositioning. So I told that in the beginning, there are many areas. In silico approaches are there, mixed approaches are there, knowledge approaches are there. So many approaches I mentioned that. So this is a typical area. There is no role of medicinal chemist. So you see here, this particular a study was established in a British medical journal in the year of 2000. So they established a clear factor that more than 70% of the patients which are consuming the metformin with a lower risk of breast cancer. You see here, we are employing or we are administering the metformin for treating the type 2 diabetics, and unexpectedly we show a clinical fact that there may be a chance to lower risk of breast cancer. So now they slowly started the reposition of metformin. Unfortunately, it is not came in the market. It is in the clinical trial itself. I will give the details. This is details. Now it is in the clinical third stage. I think you go for the clinical.gov. Basically, it is a US database. You can go for that. You will get the details clearly. This is now it is a third phase clinical stage for establishing the metformin. And majorly, this is, did you know this abbreviation CCTG? These are the areas, sorry, the groups which are concerning, that is Canadian Cancer Trial Groups, and NCA means National Cancer Center in the, let's say that in USA. These are the peoples we are majorly concentrating. I think 2022 March, I think the end of the clinical trial, and we can expect some good results if it is promising. Okay, that is a knowledge based, the recent knowledge based, it will comes in the third stage. Now the dose adjustment, that is also very important. Okay, very interesting. I think, can you see the this particular slides? Can you see the slides?
Yes, sir.
Yes.
Okay, I think the very famous structure, methotrexate. I think especially if we are concerning about the synthesis, I think you know that final year medicinal chemistry people, very difficult synthesis from the peptidine system, glutamic acid, very complicated synthesis, very big structure. Basically, it is a folate reductase antagonist. You know the mechanism. Okay, I'm not going for the detailed pharmacology. Okay, you see here, this is a methotrexate. You know that basically it is used for the treatment of tumors or something cancerous. So it's, you see here, the heading is dose adjustment also can make the drug reposition. Now you see here the folate antagonist. Okay, this is high dose. It can be an anti-cancer. In the year of 1950, this particular drug was established, entered in the market. You see here the same drug molecule. Can you see a different slides? Can you see that? The same drug molecule, 2.5 milligram. Can you see the therapeutic indication? Can you see the therapeutic indication? Hello?
Yes, sir. Yeah.
Yeah, the therapeutic indication, you see here, one is used for cancer and another is psoriasis and rheumatoid arthritis. Can you see in a normal human being, can you see any relation between cancer and the RA? RA means, I'm using the short form because that is rheumatoid arthritis. You cannot, in a common people, you cannot see any relation. So methotrexate, commonly we know a established mechanism. We studied the established mechanism from the pharmacology. It's a folate antagonist, so it can maybe cytotoxic and many things related with the pharmacology we studied. You see here, this is a dose level, but if we are changing the dose level, you see here, it shows an entirely different area. It can be used. Then we will see that in that particular case. That's why I told you, that you should not about a disease to a disease relation. You should not that. That's why. And while going for the disease to disease relation, you should go to establish a disease to gene, disease to target, disease to gene expression. Okay, I will show the hidden mechanism. MTX normally we know that methotrexate. I'm always using the short form, that is folate antagonism. And also there are some more areas of targeting, adenosine signaling, ROS generation. ROS, we know that reactive oxygen species. So commonly we are employing, because my drug designs are ROS generation types of signaling. Okay, so ROS generation, these type of areas we can see that. And now you see here, we know only the MDX having this folate antagonist, but there may be another type of mechanisms are also there. In that, what is the thing is lower dose of MTX increase the adenosine, and that may be the therapeutic indication of your RA. So here, by the identification of the pathophysiology between the two diseases, will clearly tells about that, because there are many mechanisms can be many detailed mechanism is available. You go to the cancer research journal in the elsewhere, you will get that detailed mechanism how it may happens in a low dose level from the methotrexate to its psoriasis and the rheumatoid arthritis cases, how they are effectively or how they are beautifully executed this particular drug. Okay, I think sorry for the time which I took. Okay, now we are going to the last area of my slides. So these are the challenges of the drug repositioning. Okay, the challenges, major challenges: make use of a massive amount of medical data. The medical data are massive amount we need for a clear drug repositioning. Without a massive medical data, you cannot employ a particular drug molecule. And this is a most serious problem. Do you know what is meant by intellectual property issues or patent issue? Suppose if you are designing and synthesizing a molecule, suppose if you got a patent for a particular molecule, okay, so in every patent have their own rules are there. Suppose if the metformin, if it is established for a type 2 diabetics, so if I want to use the same drug molecules for another issues, there may be a huge amount of intellectual property issues are there. So that intellectual property, you should solve between the two partners. Okay, so that intellectual property issues is a common problem, it will arise. But anyway, if you are giving the money, every problem will be solved. The intellectual property issue also can be solved by giving the money. There is no issue in that. But anyway, in a practical challenges, I just included that intellectual property issue is also one of the challenging problem. Now the machine learning, this is a concept I told that we need in the pharmacy profession. We don't have the machine learning algorithms. So that type of algorithms, we should know that. Please go to the drug repositioning algorithms. Articles are there. We can see that the basic concept of algorithm what they employed. And still is a risky business. We know that suppose if you're lucky, you will get millionaire. Okay, so it is a risky business. So these are the simple concept of drug repositioning of okay, you see, I think Prashant sir, can I continue 5 more minutes?
Please sir, please continue sir, it's very interesting.
Okay, now we will see the knowledge based screening. This is a simple concept. Okay, and what we are using in our lab, which is entirely different. Basically, it is obtained from the database of drug discovery. Okay, this is a knowledge-based approaches and screening-based approaches. Okay, I think the graphical representation is enough idea to explain. Okay, this is the last part. Okay, so yeah, many times I'm telling this is the last part. Okay, sorry for the trouble. Okay, this is a hands-on training. I will show that how the repositioning can becomes by use of a software. Are you interested? Anyone have any problem with that? Are you interested?
Yes, sir. I think all of the participants interested.
Yeah. This is a very famous software. This is a commercial software. Suppose if you are going for in Google, you will get the platform, but unfortunately you will never get the prediction. It's have a you should take the separate license for that. So fortunately, I got a chance to work with this particular professor Nicolotti. He's my collaborator in the Italy. So he only developed this particular software in the year of 2019. Which is published in Chemical Information and Modeling in ACS publication, American Chemical Society. So personally, he gave me this particular, because his area of research is drug repositioning. So if any people are interested in this, you can join them with a clear proposal. So we will admit that, no issue in that. Okay, now, okay, this is a basic concept of our software of Musel. Okay, I think, can you see this? How many drug molecules, sorry, small molecules are available? More than 18 lakhs of small molecules. So in that, there may be a 32% can be probability, can be predicted. Okay, so this is also an activity prediction. And behind the ski, okay, this is general principles. Okay, no need to go for much detail about that. Okay, you see here, this is our publication of European Journal of Medicinal Chemistry of the drug repositioning. Okay, I think in this particular area, you can see plenty of drug reposition techniques in a detailed way. That's why it's a very difficult to establish. Can you see the slide clearly, Prashant sir?
Yes, sir, it's visible.
Okay, so you please go to this particular full text. So go to the full text and you will get a detailed idea about how we executed the drug repositioning, various approaches we employed, and we will show the recent some of the publication. I showed that how the reposition can becomes in our own molecule itself with the help of these mucil. And this is a very latest publication, the Molecule Switzerland Journal, and we also employed with the help of this particular scientist, we established the details of our drug repositioning. Okay, can I slides? Anyway, thank you for all the participation. Thank you for the patience of hearing. Okay, we will. I will show the demonstration of that particular software.
Good morning, sir.
Good morning. Please introduce yourself.
Yes, I'm Mohammad Eki Azad from Bangladesh. Sir, actually, I want. Sir,
You're?
I'm Mohammad Eki Azad from Bangladesh.
Bangladesh. Okay.
Yes.
Tell me.
Sir, can I ask you question related pharmacodynamics? That is, you already discussed about methotrexate also used in psoriasis and arthritis. You already mentioned that. Should we just prescribe or ask to use any folic acid supplement? Or another question related to that, the methotrexate can cross the blood-brain barrier and it can cause chemo fog, that is brain toxicity.
I didn't get your question. Please tell the.
It cross the blood-brain barrier and cause the chemo.
You see here, you see here, blood-brain barrier prediction and blood-brain barrier crossing. So it should be target only where your drug molecules acting on the CNS. Okay, so I will tell a very simple example. Suppose if you are making a drug molecule with a good target in the acetylcholinesterase, which is typically acting on the CNS. So if your drug molecules doesn't have enough lipophilic units in your structure, so it will never cross your blood-brain barrier. So at that particular time, very famous example is dopamine. In the case of your dopamine, if you are employing a particular drug molecule, so basically it is not that much of lipophilic. So many hydrophilic units are there. So at that time, protein can be employed. So your question is, so is there any chance for the blood-brain barrier prediction? Is there? That is a question, you know. Hello?
Yes, sir. Yeah. How many ways we can just minimize the toxicity? It is the related question to your topic?
Yeah, because blood-brain barrier prediction also. So suppose if it is crossing the blood-brain barrier also, so you can predict that. I will show one software is there, SwissADME prediction. So from that SwissADME prediction, you can analyze that whether it may be cross your blood-brain barrier and whether it may be PGP substrate or that. So that is nowadays it is not a easy one. Sorry, not a difficult one to establish. What is that? These predictions. So predictions are very easy for that. Please note that its software name is SwissADME prediction or Swiss target prediction. It is available for your blood-brain barrier prediction. So I don't know about the clinical features, how it will be, if the methotrexate will pass on the blood-brain barrier, is it will affect or the RA or not. Clinically, I don't know that. Clinical information.
Sir, in our country, actually, my relatives is suffering from psoriasis, and he is already prescribed by methotrexate, but the doctor didn't prescribe any folic acid supplement without any duration. That is, at the same time, folic acid and methotrexate cannot be used, we know, but in later on, folic acid supplement should be prescribed or not, sir?
Actually, regarding that prescription, dear, I don't know exactly. Because you are telling that what type of prescription can be used along with that. I don't have an idea. I will check and tell you. So commonly, the methotrexate MTX can be employed for the RA. So because due to the interference of the adenosine mechanism. So that is a basic concept I am going to explore.
Yeah. Sir, we have seen that many of the drugs have been rejected by FDA. Those drugs which have been rejected by the FDA, can we take them and find out?
That's why I told you.
That
thalidomide is a very clear example, and we can use also fenformin is a rejected one in a USA. Fenformin. Nowadays, my area of research is fenformin. Because don't think that rejection is in a one sense it is okay only. Maybe it may be not in a good in clinical trial, but in many cases you can do the molecular hybridization. You can see the change in the functional group. So many things you can see. We can work out for a withdrawal drug also. There is no issue in that. So, but you should know the chemistry of the molecule detailedly. So anyway, the work is the same workflow only. The concept is what is the new idea you are executing. That's all. Withdrawn drug also, there is no problem.
Okay, sir.
Any more doubts? Hello, participants. Hello.
Hello, sir. Can you hear me? Can I have a question?
Yeah. What is your name?
So myself Prashant. My question is that what is the
which college?
Sir, I am from Amrita School of Pharmacy.
Okay, you're a student or a teacher?
I'm a PhD scholar come faculty.
Oh, you're a PhD student in chemistry.
Yes, sir.
Tell me.
Sir, your presentation was good. Actually, my doubt is that what is a normal duration for a drug repurpose drug to come to a market? Unlike the
2 to 3. 2 to 3
years. Okay.
And also, Prashant, what is my core concept is, suppose if you are, because that's a sorry Prashant, because actually if I will show this software, obviously I will show the chances of drug repositioning the flavipinavar itself. So in the software itself predict that there is a chance for the carbonic anhydrase 7 inhibition is there. Because recently I am working on that particular area, but unfortunately I don't know what will happen my software. Sorry for the trouble, because if I will show that, it will be very useful, and you will have, suppose if you have a drug design idea, you can enlarge your drug design concept. But I think maybe the server problem. Suppose if any person, so on personally, I will mail you the the license of the software. No issue for that. Okay, I don't know. Okay, and one more thing that Prashant, what I am expecting. Suppose if you are going for a drug repositioning, so normally that's why in the last slide I mentioned that before the drug reposition, suppose if you didn't get the exact biological scenario or biological profile, so you can see some slight molecular hybridization in that repurposed molecules. Maybe sometimes. So last time we published one of the very famous article in the the alteration of metformin molecule. Okay, we got very successful results. Now it is under the patent way, but that's why suppose if it is a molecular hybridization at the end of your drug reposition results also, that is also highly appreciable. The major thing is how we are executing the functional group with the convenient of your drug receptor interaction. Okay, so that is one of the very important things. So majorly it will take 2 to 3 years for the market. Okay, what is your area of research, Prashant?
Sir, I am working in nanotechnology approach for the autoimmune diseases.
Okay, so you are not touching with the synthesis and design.
Sir, my post-graduation was in synthesis. Right now, I'm fully in nanomaterial synthesis.
Okay.
Thank you, sir,
all
for
the
that.
best, dear.
Okay, sir. Thank you for the answer.
Any more doubts?
So can we wind up, sir?
Yeah. I think, sorry for the trouble.
Yeah. As the participants don't have any questions, I'll wind up the session. Thank you, Bijo sir, for an informative session on also giving some practical approaches on drug repositioning. And also, I thank you for providing answers to all these questions. And I would like to thank you again for joining us in this seminar and for providing all for the drug repositioning and drug discovery. Once again, on behalf of Department of Pharmaceutical Chemistry, Nirmala College of Pharmacy, our heartfelt gratitude to you. Thank you very much, sir. I also thank all the participants. I hope that you all have gained some information from this webinar and also got inspired beyond the technical glitches. We also will be sharing the YouTube video soon, and the feedback forms will be sent to you to your registered emails, and also your certificates after filling the form. Thank you once again. Thank you all.